big question is, can we make medicine for endangered species? Can we make precision medicine for endangered species is our dream. And so I've been traveling through Africa trying to answer this question, what is conservation chemistry? I have so many new ideas for the past couple days I've been here. And so my talk will be broken into two parts, which is kind of follows my life. That the beginning was in drug discovery, and I'm very engaged in drug discovery. I work with a lot of pharma companies, consulting now, and I have historically. And then how can we use that to impact environmental health is hopefully where we'll get to. For 20 years I've been working towards this dream that you could design, make, test a drug, right? That one day we're going to have a big button that you can push, and you could, say like my grandma has pancreatic cancer, and I want to treat that cancer, and I need a medicine, and we're going to get better and better at making medicines for these targeted approaches, and events like this are really accelerating this. And so this is a dream. This is not my dream. This is the dream of a multi-trillion-dollar pharmaceutical industry that is pushing towards this accelerated invention of medicine. There's been a lot of data that goes into this, and so systems biology is I'd say an area that is really well established and has lots of data that goes into it. And so you guys are digesting data right now and navigating data, and it's quite tricky, right? But so what we might do is go to our patient, get some tissue, get some cells or some blood, and do a series of analyses on these tissues, looking at genomics, transcriptomics, metabolomics, proteomics, and it's a lot of data that comes out. But hopefully at the end you can digest that data and arrive at a pathway or a protein that is associated with the disease, and then a chemist can come in and say, I'm going to make a molecule that sticks to that protein and treats that disease. When you swallow a pill, it's typically a molecule that sticks to a specific protein associated with the disease, right? And so my career is as a chemist. I like to think about molecules. What are molecules? Where do they come from? And so that's this other side of all of these building blocks and transformations, reaction conditions. And so we've been using AI to accelerate this. The picture kind of looks like this. You have this big blob of the blue thing is the light blue thing is a protein. That's what's causing the disease. And we need the yellow thing, the little yellow molecule that sticks inside. It sits in the active site of that protein, and it stops the disease from happening. And then there's other molecules that we could consider, but they don't stick to the protein. They're not good, right? So we want to filter to the good ones, not to the bad ones. The beauty is that we have really nice generative tools for protein structures. Right now, we can think about them like movies, and we can generate the impact of this. I don't know how many people here are thinking about molecules in their day-to-day, but so I just want to introduce a few ways to think about molecules that are important for doing this type of research. And so I show you three different representations of the drug Tamiflu. We're very worried about avian flu in North America right now, and so I talk a lot about this molecule right now. And so you can see that there's a line and stick drawing up at the top. If you've ever taken an organic chemistry class, maybe you've seen that. I love drawing hexagons like this all day. I can just sit at the chalkboard and draw such things. It's got a name, oseltamivir, or it's got a brand name as Tamiflu. And then the bottom one there is what's called a smile string. And so if you've seen this, great. If you haven't seen it, then it's a way to communicate that chemical structure. If I take that string of text, it describes the exact same molecule up top. There's another way to think of these molecules that's really relevant to me. One, there's two definitions here. One is that it's as a graph. Okay, so hopefully you can see that the bottom structure there is the same one as the top. It's got, but rather than the carbon and nitrogen, there is nodes and edges. And then another important component to this is that it's being built from smaller pieces. This is where medicine comes from, right? Or synthetic medicine anyways, that you have pieces and Lego blocks and you click them together to get the structure that you want. Often I would talk a lot about our work in digesting graphs to optimize synthesis. I'll just quickly show you that this is a very short synthesis of a natural product we were after, and we used graphs to get there. And I want to highlight here these reaction conditions. These are kind of a difficult part of the recipe that we need to get to. I'm bringing this up just because I wanted to talk about some of our work with LLMs. And so to identify these reaction conditions, we have to look through a lot of possibilities. There's like 10 to the 70 possibilities that I could go into my lab and make a mixture of different reaction conditions any day. So this is pretty antiquated. This is from February of 2023. So this is like kind of one of our first prompts in ChatGPT. It's become a lot more sophisticated. But so we're just telling ChatGPT, generate a 3 ligand by 4 catalyst by 2 reaction array to run this type of reaction. And this is the output. And so now what you see is that I have a series of reagents that have different reagent classes. And I can go into my lab and I can mix them. And we do that. And I think one of the reasons that I like to show this to this audience is that I find that LLMs are really good at giving an approximation, right? But they're not good at giving the exact answer. And so we have a tool that builds these arrays. What you're looking at, all the pink and purple dots, each square is a different chemical reaction that we ran. And there's one of them that's really good. And so it's like ChatGPT, when we asked her to give us this recipe, she gave like a good approximation, but not a good exact answer. And so there were, by building this array around that, we get a kind of a sampling of the space around where we were asked to look. And there's only one really good answer within there. But okay, so we're good at making molecules. We're good at designing molecules. We design, make, test, and we can make molecules really quickly. Here's a particular selective cancer molecule that we've made for human patients. These are human cancer cell lines, and we used all of our AI tools to generate this particular molecule, and it's very exquisitely shutting down cell cycle arrest in these cells, which is what's shown here. There's a lot of exciting ways to leverage such information for medicine. And again, the pharmaceutical industry, I think, is really pushing towards this, button that you could get a drug for any disease. And so the question we asked is, what if our patient wasn't a human? So what if we then approached the situation and said, what if we encountered a health situation that was, not a human? So these are some of the patients that we're currently working with in my lab. They do not have good health care. They don't obey all of the biological rules of what we know about human medicine, but there's a lot of things that do overlap. And I guess I spent a lot of time. I did this out of a passion for conservation, but it's like should we really invest resources in saving a penguin? I don't know, but. This notion of One Health, I think, is one that hopefully will resonate around this room. And so this is a really fascinating paper that came out late last year from Eyal Frank in Chicago. And so one of the diseases that we work on is called white nose fungus, or white nose syndrome in bats. Bats in North America contract this fungus and they die very rapidly. So it's a really big problem for us. We're losing our bats. And what Professor Frank has done is looked in the hospital records where this disease was reported. And what he noticed is that there was an 8% increase in human infant mortality. Okay? So, we were gung-ho on making drugs for bats because we thought bats were cool. We wanted to save them. And then this paper came out. It's like, this is like an 8% increase in infant mortality when bats contract this disease. You start to unpack it, and what is the ecosystem of a bat? Ecosystem service of a bat, right? Is to remove insects, and so when they disappear, there's more insects, right? The farmers spray more pesticides on their crops to deal with those insects, and it's well known that pesticides correlate with infant mortality, and so it's kind of this direct, connection of human health to ecosystem health. All right, so let's go to Florida and take a look at some of the work that we're doing with the Loggerhead Marine Life Center there. They unfortunately pull about 5 sea turtles a week out of the ocean in this condition. They are battling a infectious cancer, and this is now the major stranding event for these sea turtles in Florida. Here's Bellatrix is a sea turtle who's about to undergo a procedure to have these tumors removed. But what you can see is that she's just covered in cancerous lesions, right? It's gross, I'm sorry, but it's, and so what you see on the right is a drug target that we have pulled out from sea turtle cancers. And now using AlphaFold, which is a new tool that you can generate a protein structure, gives us this 3-dimensional picture that we can actually start to look and get in and start doing drug discovery. And so we have an incredible arsenal of cancer drugs available to us today. And so we find it fascinating that that these sea turtles are battling cancer at a time when we have access to a lot of new wisdom around cancer medication. I'm going to take you now to the northeastern forests up around where I live, and this was actually our first patient was a tree. We started working in trees, and it was an interesting decision to start in trees. But this one in particular is really important to our ecosystem, and I want to share why. So it's called the hemlock tree. It's the state tree of Pennsylvania, as one of our states. And it carries this really important ecosystem service. So this is a photo from the same place in Michigan in the summer and the winter. And our forests are, they have a dense canopy of leaves like you see here. But when the winter comes, all the leaves fall down, you see? And it kind of gets covered in snow. It's lovely. But this tree is a conifer that can live in the shade. It's the only conifer that we have in North America that lives in a shaded environment. Normally we need bright sun for our conifers, like your pine trees or a spruce or a cedar tree. This one particular tree can grow in the shade, so it can grow under the canopy of the forest. And you can see why that's important in the winter, because the trees drop all their leaves. And so now we have some cover for our animals throughout the winter. These are really, really critical trees to our ecosystem. Unfortunately, we have lost a very large number of these trees to an invasive insect. So there's an insect called the hemlock woolly adelgid that came in from Asia, and it has wiped out this tree in most areas where it is. It's only just recently reached us in Michigan. And so this is one of our important national parks. This is the Great Smoky Mountains, and every tree that you see that is living is not a hemlock tree, and every tree that is a hemlock tree is dead. And so this is a big problem. Here's the insect, and it creates this kind of white waxy coating around itself. It like sucks the blood out of the tree and turns it into this white wax. And so we thought that might be important. You can see the spread of the insect. It was introduced to North Carolina on someone's bonsai tree in 1951, and you can see it's just spreading across the country. So we're looking at this through the lens of tackling it through its white wax producing mechanism. But I want to introduce you to another insect of our forest. This is the boogie-woogie aphid that is also a white wax-producing insect that's kind of wiggling around. My daughter hates them. I'm always like gathering them from the woods, but my 8-year-old daughter is very angry whenever I'm like, oh, come look, there's boogie-woogie aphids over here. But what we would like to do is selectively target this hemlock woolly adelgid while not impacting this boogie-woogie aphid that has an ecosystem service that it plays. And so we've recently sequenced the genome of the invasive insect, and one of the proteins that we intend to go after is shown wiggling here, which we can generate these structures using new generative AI techniques like AlphaFold. So we'll go now to Panama and all through Central America. The frogs of Central America are in a really bad way. I went up to the Aberdares, or the Aberdare Park. It's the most beautiful place. It's incredible up there, what you guys have. And I told the game warden I want to see frogs, and he laughed at me. Apparently you don't go to the Aberdare to look at frogs. But I'm very interested in frogs. We are unfortunately living through a mass extinction event for all amphibians on this planet, and so we've lost about 400 species of frogs to an invasive fungus called chytrid, and so these are some frogs in California. I had some fun looking around at some of your native frogs. So these are all frogs from Kenya, and that are, it's kind of this really beautiful diversity. Maybe the colors don't show up so great, but there were hundreds of Kenyan frogs that were just like beautiful to look at. Except the yellow one here is actually not from Kenya. This is the Kahansi spray toad, which is from Tanzania, and it is extinct in the wild. And so it is being preserved within zoos, but through this fungus, and also through habitat loss, this poor little frog is on its way out. And if you think about what I said earlier about the bats, right, that when the bats get a disease, infant mortality goes up. What is the ecosystem service of a frog, right? They remove insects from the ecosystem, and so, it's maybe hard to think of a frog as an important patient, but they really are a critical part of our ecosystems that we need to respect. Now, luckily, one of my dear friends and partners on this mission is Tim James, and he had developed a really nice model system using these African dwarf frogs. Does this frog look familiar? It's an African dwarf frog, so I don't know if I'm in Africa, so it's just always been this interesting. But anyways, they're widely available in the pet trade, and the interesting thing is that they're easy to infect with chytrid fungus, and so we have this system here where this is loaded up with the 400 frogs, and we can do all types of experiments to look at them. Here's some of them swimming around. I guess it's not really showing up great, but they're tiny little frogs, and they're easy to breed in an aquarium setting, and so it allows us to very quickly take our findings from the lab and do clinical trials in this model system. What that looks like is using some of the drug discovery tools that I mentioned earlier to find antifungals. And what you're looking at here are dose response curves. And there's two drugs, or actually three drugs, that are like right at the bottom. Those are incredibly potent antifungals for killing this chytrid fungus, like incredibly potent. And so we're hopeful that we can use that as a tool to mitigate this situation. And working in African dwarf frogs, as a model to these two frogs from Panama, one is the Panamanian golden frog, the other is the lemur leaf frog. This is work that we do in partnership with the Smithsonian. Brian Gratwick is a leader of Panamanian golden frog rewilding. But the problem is that the frogs just get out there and then they rapidly die from the fungus. And so we're trying to work with them and understand how we can bolster their immunity and provide treatment options. All right, and I want to end with a story that takes us. It's actually happening all in Ann Arbor, Michigan, where I come from. But the animal is a Gila monster, which is from the deserts of Arizona and northern Mexico. And so this is a really fascinating story. We were at a zoo doing our holiday party and talking about frogs and turtles and some of this work that we were very interested in. And the zookeeper said, you're talking about this wildlife disease. We have a Gila monster that we are about to euthanize because he's got cryptosporidium. It's a parasite that there's nothing that we can treat this animal for. Now, as a pharmacist, this really resonated with me because the Gila monster is where we got the drug Ozempic from, or at least the saliva. It was the first generation of this weight loss and anti-diabetes drug Ozempic. And I don't know if you guys are familiar with this drug, but it's completely changing the game of diabetes treatment around the globe, and is raising billions of dollars. And so they're selling like $4 billion of Ozempic a quarter. And yet the animal to which we owe some debt of gratitude for this drug is about to be euthanized because it's got a disease that there's no treatment for. So we got interested in working with Pebbles, the Gila monster, Here's Pebbles, and she was battling Cryptosporidium. She needed to be kept separate from all the other reptiles in the collection, and it's really quite challenging when you bring in a reptile that's got Cryptosporidium, because it could infect a whole collection if you have that. So here's Dr. Mohammed Ishfaq working with the team at Creature Conservancy, extracting some of the stomach contents of Pebbles, and we put those under the microscope, and it's just bizarre. It kind of becomes apparent why it's so darn hard to kill this thing, because it's got this really protective shell around it in panel A. In panel B, this is a different life cycle of the parasite, and you see it's got this kind of egg-like coating around it, and so this is just very, very well protected. It's very difficult to work with this, but we did use LLMs to solve this problem, and I wanted to show this tool that we're building. It's quite early in its development, but it plugs into a different component called Semantic Scholar. I don't know if that is familiar to anyone here, but Semantic Scholar is like a library networking tool, and so it will tell you what papers cite other papers. And so this has been really beneficial to us, because what we can do is take 5 papers that we find on Google, just do a Google search for 5 papers, drop them into Semantic Scholar, and then it will build a whole landscape of this paper cites these papers cite these papers, and then you get 100 papers or 300 papers and kind of get all the information about a field of research, right? So now we've got hundreds of papers related to Cryptosporidium, and then we use ChatGPT to read them and identify drug names. And so from those drug names, we built a simple histogram like this. We've been doing more to inject context into this. These are context independent, just is the drug described in the paper, yes or no? But what you can see is there's 2 drugs that are really the outliers. One is paromomycin and one is nitazoxanide, and we went with paromomycin for reasons I can explain if you're interested. And we were happy that we did. And so here is Cryptosporidium from Pebbles that has been treated with paromomycin. And what you see is these big white kind of crystalline occlusions. We never saw that without treating. So this drug is harming this parasite in some way. As well, I don't know how easy it is to see on the slide, but that kind of really thick, non-penetrable layer is getting a little bit busted up around the edges, and so we feel good that this drug is doing something. So then with our veterinary team, the drug was formulated into quail and fed to Pebbles, and she got 8 grams of this drug over the course of 2 weeks. And I'm excited to report that our first test on her was last week after treatment, and she's negative for Cryptosporidium for right now. We need to continue testing and hope that we are there. But as well, the zookeeper says that her appetite has come back, and so hopefully LLMs were helpful in making this intervention. In trying to work with an endangered species that was slated for death, right? I mean, when they realized the animal was sick, the plan was to euthanize, to get rid of it, and yet, we owe a multi-billion-dollar pharmaceutical market to this creature, and so I think it was a really fun way to use LLMs to pull in a lot of information. And so, as Jeffrey described, we're very excited about this new direction of taking all of the things that have been developing for human medicine, which have come a long way, right? In the same way you guys are witnessing LLMs advance, you can imagine that drug discovery is accelerating at a rapid pace. And so we imagine that one day it will be accelerated so fast that we could go beyond humans and imagine patients, regardless of how much health care they have, but really based on the ecosystem services that they provide.